Ophidiomycosis, aka snake fungal disease, and pentastome parasites on snake health. You all are in for a real treat today. So without further ado, I would like to welcome Jenna and Terry to present. Thank you.
Carrie, thank you so much. So like Carrie said, today we're going to be talking about snake pentastomes in North America with a particular focus on the invasive species that is causing problems in our native snake populations. So this species is pictured here moving and being pulled out of two rattlesnake species. So on the top left, we have our pygmy rattlesnakes that we've been talking about. And on the top right, we have an eastern diamondback rattlesnake. So we are invested in researching the invasive parasite, the invasive pentastome parasite, and its impact on native snakes for several reasons. First, there is evidence for clinical signs of disease and mortality related to infection. And second, the parasite utilizes many hosts, both in its immature and adult forms. And third, there's no known limitation to its spread in North America. So before we get started, I just want to clarify some terms and definitions and make sure that we're all on the same page because we're going to use these terms throughout. So pathogens are disease-causing organisms, and these can be parasites like nematodes, fungi like the pathogen that causes snake fungal disease, and protists like Purkinsia that infects amphibians. Parasites rely on their hosts for sustenance, but not all of them are going to cause disease. And it's hard to know clear details of infections and associated disease without performing experimental infections. From what we do understand, the invasive pentastome parasite that we will talk about today does cause disease, and this is known as pentastomiasis. We'll talk further about the clinical signs of the disease later on. But before we get there, we just got to talk about a few more terms related to the complex life cycles of parasites. So these are the terms we use to define different types of hosts in the complex life cycles. The definitive host is the host where the parasite develops into a mature form and reproduces. This example on the right is a life cycle of a trematode, and we can see that after ingestion of an infected prey item, the definitive host, the heron, becomes infected, and soon the trematodes develop into adults and produce eggs. Those eggs are then released in the feces, which are then ingested by the first intermediate host. The intermediate hosts, the eggs develop into an infectious larval form to then infect a second intermediate host. And a paratenic host is different than an intermediate host because it allows the immature or larval forms to persist but not develop. And all of these types of hosts play a role in snake pentastome life cycles. So our focus today is obviously the snake pentastomes, and they do have complex life cycles relying on at least one intermediate host. Our focal species, or the invasive species, seems to exhibit a three-host life cycle, like we see here with the trematode. So just a quick overview of our topics for today. We're going to cover a brief backgrounds of pentastomes and the native and invasive snake pentastome species here in North America. And we'll pause for a Q&A prior to getting into conservation concerns and future research. And then we'll close with a second Q&A. So pictured here is a southern black racer from Florida with multiple invasive pentastomes crawling out of its mouth. And I would just like to note that gloves are an important part of reducing the transmission of pathogens. And this snake was outside of my driveway in Orlando, Florida, so I acted rather quickly. So despite my poor biosecurity practice or lack thereof, it is important to wear gloves to reduce transmission between animals and when dealing with pathogens that we don't understand the zoonotic potential of, so to inhibit transmission to yourself. And we'll get into zoonosis in a moment. So with pentastomes, there are a few species known to infect humans, but the concern of zoonosis or human infection from infected wildlife with the invasive pentastome is currently unknown, but it's important to be safe. So we've been chatting about these pentastomes. So what exactly are pentastomes? They are endoparasitic crustaceans. So they reside in the upper respiratory tracts of carnivorous reptiles. Mostly, besides a couple that infect mammals and birds, if you don't consider birds reptiles. And just to remind everyone, just where crustaceans lie in the tree of life, Crustacea is a subphylum of arthropods, and the phylum of arthropods is usually characterized by chitinous exoskeletons, segmented bodies, and paired jointed appendages, all of which pentastomes have. So as obligate parasites, these pentastomes or pentastomes at large are hematophagous in the respiratory systems, so they're feeding on the blood. So on the right here, you can see a mix of penistome adults from 9 different genera to get an idea of the morphological diversity that penistomes exhibit. Interestingly, penistomes are the oldest parasite known to science, which is really cool, with fossil records dating to the late Cambrian period. So that's about 150 million years prior to their terrestrial vertebrate hosts' arrival. So at some point in their evolution, they were free living. So these parasites have really been misunderstood as they have a mixture of shared characteristics between arthropods and annelids. They even have a misnomer, so they're called pentastomes, which means 5 mouths, but they actually only have one feeding apparatus, which you can see in the anterior portion of the heads here. I'm not sure if you can see my cursor, but I'm circling around it. And that's really their only feeding apparatus. And the other 4 mistaken mouths are actually hooked-like appendages. Much of their taxonomy in the past has been confusing with strictly morphological and developmental characters studied prior to current molecular tools that we now have. But with those tools, we now understand them as a class of crustaceans. So this class of crustaceans is most closely related to maxillopods, which includes Branchiera, which are fish lice pictured here on the bottom left, and remipeds, which are tiny free-living venomous crustaceans. So if you're interested in further details about the evolution and the phylogeny of penistomes, visit the Wakelet that Carrie will share in the chat. It has a bunch of resources. But currently myself, Dr. Terry Farrell, and Dr. Robert Fitak at UCF are creating genomic resources to hopefully clarify their evolution. They kind of occupy like a main hanging lungworm hanging out of its mouth. And on the left is an adult female after a couple of months. The adults can be over 100 millimeters long, so they get quite large. And we're going to see some pretty gross pictures coming up, so I'm warning you. So here are 3 different individual infections. The 2 on the left are banded water snakes from Southwest Florida. Our collaborators at Florida Gulf Coast University collected these, Dr. Andrew Durso specifically. And on the right is a black racer infection. You can see that a lot of these individuals are quite large. They get bigger in native hosts. So the pentastomes get bigger in native hosts than they do in Burmese pythons. And we also see in native hosts that the infections are more female dominant. So a lot of the larger ones we're looking at here are females. So currently we know of 17 native snake species that serve as definitive hosts for snake lungworm, and often infections are much more severe in native snake hosts compared to the Burmese pythons, with over 100 individuals feeding on the snake's blood in the lung in some cases. We know that tokay geckos and tegu lizards can also host adults as well, and there's some evidence that iguanas might too. The abundance of native and invasive definitive host continue geographic spread. So in addition to wild animals spreading the disease, captive animals can as well. For instance, many tegus are held in outdoor enclosures, and snakes are too, and that could contribute to the spread. So if those animals are held outside and they're infected, they pass eggs with their feces, a cockroach or a beetle moves in and out of the enclosure and therefore spreads the disease into the wild. So here are the documented snakes in Florida with infections. The ones in red are more recently found and not published just yet. And note that the federally protected eastern indigo snake has been found with infections, and there's further evidence. Dr. James Bogan, who I believe is on the call, is doing a lot of work looking at the prevalence in indigo snakes in Florida. And I think we're in addition to the tegus, who can serve as an intermediate host and a definitive host. So Madison Harmon, who is a graduate student at the University of Florida, has done a lot of research on penistomes and tegus and found that they can support larval growth as well as reproductive adults. And on the Wakelet, there's a link to her ResearchGate, and she's done a lot of presentations on her research. So go check that out if you're interested in how penistomes and other parasites are infecting tegus. So time for a break and to hear your questions, hopefully answer them. And we'll cover the known geographic range of RO and the likely conservation concerns after this Q&A. concern. So when snakes ingest an infected lizard or frog, the lungworm larvae bore through the gut and migrate to the lungs, where they encyst in. Experimental infection studies, which the title was just right there, have demonstrated that anurans in lizards are prominent intermediate hosts, and this corresponds with the diets of the snakes that are most often infected. There are, however, snakes that are infected who mostly feed on mammals like eastern diamondback rattlesnakes. And currently, unfortunately, the only record of a Raleightiella from a mammalian intermediate host was a Kenyan shrew. So mammals play a role in this life cycle. We just are unsure of what it is exactly. So our experimental infection studies support that this lungworm likely exhibits a 3-host cycle, starting with an invertebrate. And the lungworm's eggs are passed with the feces of the snake, which are then ingested by a coprophagous invertebrate, so one that eats feces like a beetle or cockroach. And on the top right here, you can see pictures of RO eggs from a snake's fecal sample. And the infection studies are on the Wakelet as well, so you can check those out. So this is what larvae removed from the fat bodies of the infected roaches look like. So that's where they migrate and encyst. They're quite small at this stage, about 0.2 millimeters, and they're not visible to the naked eye, but they do still, however, sport those 4 hook-like appendages that we've been talking about. So when a lizard or a frog consume an infected cockroach, they became infected with larvae about double in size. And here you can see those larger larvae through the body wall of an experimentally exposed brown anole. From the experimental infection studies, again, and you can find those on the Wakelet, we did not find impacts of RO infection on the growth rate or survival of the intermediate hosts. And though this makes sense given the size of the larvae and the relative inactivity, more research is definitely needed to confirm this and whether or not RO is a concern for intermediate hosts as well, a conservation concern. So when snakes ingest an infected lizard or frog, the Parasite crotali uses mammals mostly as intermediate hosts. Location of 27% of North American pit vipers with Porocephalus crotali infections. And on the right, you can see the life cycle of P. crotali and the succession of its molts within the host. And we know so much about this life cycle because in the 1960s, there were some experimental infections with cottonmouths and rodents. From what we know, Porocephalus coarctatus uses fish as intermediate hosts, but it might use other intermediate hosts as well. And we see a highest prevalence in natricines or water snakes. So though penistome infections are diet dependent based on the intermediate hosts, there are always exceptions to those being infected, especially with snakes that have a more general diet and eat a variety of hosts. Tari, do you have anything to add?
No.
To learn and know more about, and in somewhat of a row and in similar sizes, which is distinct from the invasive species that we'll talk about. So Porocephalus crotali species that we'll talk about of the snake lung, and each species has hooks aligned in some, which is on the right, which after handling infected snakes or their substrate, there is a potential for ingestion of hooked snake for all. Talk a little bit about some are focusing on today through these 3 life stages we see here. And there are many different types of penistome species. There are some that infect gators, alligators, some that infect turtles, some birds, some mammals, and many infect snakes. In all stages, they have the 4 hook-like appendages that we talked about. And on the left, you can see the first stage, which is the embryonated egg, which is released into the environment from the definitive host. In the middle is the larval form, which resembles kind of a tardigrade a little bit. And on the right is the adult form. And this adult drawing is actually one of the 3 genera that are known to infect humans. So we're going to talk a little bit about zoonotic concerns before we jump into North American pentastomes. So we don't know the full extent of zoonotic concerns for all penistome species beyond these 3 genera listed here. So Armilifer, Porocephalus, and Linguatula. Zoonosis, again, is a disease that can be transmitted from animals to humans. And the highest prevalence of human infections with pentastomes have been found in Africa, the Middle East, and South and Central America. There is a recent publication in a country, Indonesia, that farms snakes for food that warn about the potential dangers of eating undercooked snake meat because the snakes in that slaughterhouse had over 60% prevalence of pentastomes. There hasn't really been much on the zoonotic concerns of pentastome infections from handling captive and wild snakes. However, if hands are not washed after handling infected snakes or their substrate, there is a potential for ingestion of eggs and therefore larval infection in humans. We don't know much about the rail ITLids that we'll focus on today, though. And there are 2 publications related to human infections in the Wakelet that you can check out. So jumping into North American pentastomes, there are 2 abundant snake pentastomes that are native to North America. One is Chirocephalus coarctatus, which is on the left with a very bulbous head that makes it distinct from the other parasite that's native. And that one is Porocephalus crotali, which is on the right. Both of these species get quite large and they live in the anterior highly vascular part of the snake lung. And each species has hooks aligned in somewhat of a row and in similar sizes, which is distinct from the invasive species that we'll talk about. So Porocephalus crotali uses mammals mostly as intermediate hosts. Location of 27% of North American pit vipers with Porocephalus crotali infections. And on the right, you can see the life cycle of P. crotali and the succession of its molts within the host. And we know so much about this life cycle because in the 1960s, there were some experimental infections with cottonmouths and rodents. From what we know, Chirocephalus coarctatus uses fish as intermediate hosts, but it might use other intermediate hosts as well. And we see a highest prevalence in nature.
Nothing brings us together like egg
Of the disease. Further, there are no limitations to how far RO can spread, especially as it utilizes widespread synanthropic intermediate hosts. And synanthropic hosts just means they're closely related to human activities and they often hitchhike with us on our vehicles or in the plant trade and in other ways. So when they move, they bring their parasites along. Further, a lot of Florida snakes are often wild caught and sold nationally, so that can be another route of transmission. One lizard that we all know is exceptional at catching rides with humans is the brown anole, which serves as a competent intermediate host and is also pervasive throughout the Florida Panhandle, the Peninsula, Texas, as well as the coast of Georgia and South Carolina. So back to why we care so much about this parasite spreading further and getting into more native snake species. So there's evidence that for some species, the snake lungworm introduction is aligned with population declines, both temporally and spatially, and I'll talk a little bit about that later on. But like Terry was noting, we see variable lethality associated with infections between and within species. So with black racers, the one that was pictured on the second, like the overview slide, that one had ideal body condition. It was probably just nipped by a car and that's why it died, but it had 60 plus adults in the lungs. And when I say ideal body condition, it had plenty of fat reserves, it had little lesions in the lungs, and it was yolking up follicles. So it died for another reason, not from the 60 plus adult pentastomes that were in its lungs. Whereas some of our collaborators see snakes acting weird, moving lethargically, and then they find them dead the next day, and the infection intensity is nowhere near that severe. So they'll have like 5 to 10 adults in the lungs, and there's no other reason for them to have died. So there's a large variation in the responses of snakes to these infections. And in captive snakes with infections, there have been symptoms such as decreased breath rate, substantial anemia, lethargy, and reluctancy to eat reported. Some sublethal impacts that have been reported in wild snakes include emaciation, lesions of the lungs. You can see the hooks and how big and sharp they are in the bottom left there, as well as pneumonia, anemia, and sepsis. Whenever we say sublethal, though, it's still important to note that they are energetically costly impacts on the snakes. And when snakes are dealing with multiple infections, we don't really understand how co-infections with diseases like snake fungal disease are going to impact survival, growth rate, or reproductive output. So a lot of the studies that Terry has been a part of with researchers from New Jersey, Dr. Joey Gugliaro and Dr. Craig Lind, they've reported that snake fungal disease alone disrupts normal reproduction and therefore could cause population declines. They've also reported the metabolic costs of this disease. So when animals are already dealing with snake fungal disease and other diseases,
it's a very interesting situation. Population of pygmy rattlesnakes, and it's insipid. Actually has.
All right, so the snakes at the highest risk of infection are those that rely on amphibians and lizards as prey items. This is supported by the current prevalence data in the US and in Australia, as well as the infection studies that confirm frogs and lizards as competent hosts. So the southern hognose, which is a state protected species in Florida, relies on an amphibian prey base in Florida. Like so a lot of prevalence data will likely continue to be opportunistic. For live definitive hosts, we can assess infection through fecal sample screening. So these female pentastomes produce hundreds of thousands of eggs. So usually if you get an animal that's infected, like Terry was saying, you can either do a fecal, currently developing a swab protocol to detect infection. Terry and I started Worm Alliance in monitoring disease is surveillance in the pet trade and throughout the southeastern US. So Terry and I have found 4 captive infections so far, just opportunistically, 3 of which resulted in mortality just within months, a couple of months of being sold. And 2 of those that resulted in mortality, you can see here. So these are 2 garter snakes from the same breeder in Miami, but based on their physical appearance, like stubby tails, we do believe they were wild caught specimens. And also through social media, you can see the title of Terry's most recent paper at the bottom here. Terry found a captive infection in Michigan. So that was a banded water snake that spit out a pentastome. And a woman posted it on Facebook and Terry found it. So these accidental findings indicate that a lot more attention and resources are needed for pentastome research in the pet trade and beyond. Terry, do you have anything to add?
Yeah, I think one thing we might want to think about is the degree to which captive and wild animals are not well separated, and that's, I think, particularly true in the Southeast, where people may take snake bedding material and compost it, which is releasing eggs into the environment in places that have loads of roaches, which are known intermediate hosts. Other people buy brown anoles from Florida, again, known competent intermediate hosts, as feeder material for their snakes. And so we are shipping out from Florida tens of thousands, quite likely, of anoles a year, many of which are designated to be fed to snakes. And so there's a lot of avenues by which snakes and pentastomes can move around in the environment, and we should be careful not to think this is just an issue for wild snakes and that wild snakes and reptiles and amphibians are totally separate from captive populations. There's a surprising amount of interconnections.
Thank you. All right. Jack, I see that your hand is, because it will help us be able to morphologically distinguish if it's the invasive versus the native. And as we mentioned, there are unknown zoonotic concerns with this disease. So if you're handling captive or wild snakes that you don't know are infected, wash your hands and wear gloves if you have them. From what we know so far, both ethanol and bleach kill the parasites. So if you treat the substrate waste of your pet with either, you'll reduce the spread and the potential for zoonosis, especially if you're composting. And the other QR code is to the Park Biosecurity webinar, which is a more general biosecurity. It's based on amphibians, but a lot of it can be applied to pentastomes as well.
Dear Mahjong lovers, if you're experiencing memory loss and sleep loss. As a neurologist, I recommend you.
As well. Yeah, thank you so much for sticking with us. It looked like we retained, therefore spreads the disease into the wild. With the feces of the snake, which are then ingested. Florida Gulf Coast University collected these, Dr. Andrew Durso specifically. And on the right, and often infections are much more hosts that the infections are more female dominant. So a lot of the larger ones we're looking at here are females. So currently we know of 17 native snake species that serve as definitive hosts for snake lungworm, and often infections are much more severe in native snake hosts compared to the Burmese pythons, with over 100 individuals feeding on the snake's blood in the lung in some cases. We know that tokay geckos and tegu lizards can also host adults as well, and there's some evidence that iguanas might too. The abundance of native and invasive definitive host continue geographic spread. So in addition to wild animals spreading the disease, captive animals can as well. For instance, many tegus are held in outdoor enclosures, and snakes are too, and that could contribute to the spread. So if those animals are held outside and they're infected, they pass eggs with their feces, a cockroach or a beetle moves in and out of the enclosure and therefore spreads the disease into the wild. So here are the documented snakes in Florida with infections. The ones in red are more recently found and not published just yet. And note that the federally protected eastern indigo snake has been found with infections, and there's further evidence. Dr. James Bogan, who I believe is on the call, is doing a lot of work looking at the prevalence in indigo snakes in Florida. And I think we're seeing quite a bit of infections in the indigo snakes. So definitely a cause of concern. Here are the current known intermediate hosts in addition to the tegus, who can serve as an intermediate host and a definitive host. So Madison Harmon, who is a graduate student at the University of Florida, has done a lot of research on pentostomes and tegus and found that they can support larval growth as well as reproductive adults. And on the Wakelet, there's a link to her ResearchGate, and she's done a lot of presentations on her research. So go check that out if you're interested in how pentostomes and other parasites are infecting tegus. So time for a break and to hear your questions, hopefully answer them. And we'll cover the known geographic range of RO and the likely conservation concerns after this Q&A.
Wonderful. We haven't had any questions in the chat. Everybody's been listening with rapt attention to your presentation.